In the final Intro to Immunology mini video or mini lecture, we will discuss lymphocyte effector functions that mediate pathogen clearance. This includes how B cells get activated, how antibodies mediate their effects, and the effector T cell subsets that mediate pathogen clearance. So I'm going to go back to this slide, which was the end of the last lecture, in which we observed that naive T cells can be differentiated into activated effector T cells that can migrate to inflamed tissues, or helper T cells that can migrate to the B cell zone and enhance B cell activation. So these activated effector T cells can comprise several different subsets. Depending on the pathogen to which they are exposed. CD4 positive helper T cell subsets include T helper 1, T helper 2, and T helper 17 cells. And CD8 positive killer T cells can differentiate into, or CD8 positive T cells can differentiate into killer T cells. Cytotoxic T lymphocytes, or CTLs, is the other way to say that. So Th1 cells are specific for fighting off a whole variety of pathogens, including bacteria, viruses, fungi, and single-cell parasites. Th2 cells are really important for fighting off helminths, including worms, as well as participating in the wound healing response and in the allergic response. Th17 cells are really important for fighting off extracellular bacteria and fungi, and for maintaining barrier integrity at epithelial barriers. And lastly, cytotoxic T lymphocytes are killer T cells. They kill virally infected cells, and so they're critically important for the antiviral response. Can be important for some intracellular bacteria as well. T helper cells will migrate into inflamed tissues and enhance the activity of innate cells. Now, I don't want to go deep into all the different helper subsets in this introductory session. So instead, I'm just going to use Th1 cells as an example of how this works. This is a Th1 cell. It secretes a cytokine called interferon gamma. And this cytokine, when Th1 cells are interacting with macrophages in inflamed tissues, can enhance the activity of these macrophages. You'll notice that these Th1 cells are also expressing a co-stimulatory pathway where the Th1 cells express CD40 ligand and the macrophages express CD40. When these interactions occur, this dramatically enhances the activation of this macrophage. So this Th1 cytokine, interferon gamma, enhances macrophage activation, and these activated macrophages in turn have increased antimicrobial function. They're better able to take up and kill pathogens within their phagolysosome. Now, killer T cells, on the other hand, have a different function in which they directly kill virally infected cells. And so this is a cytotoxic T lymphocyte interacting with a virally infected epithelial cell and inducing its apoptosis. And the way it does that is through the secretion of these cytolytic granules. This CTL will form a cytoplasmic bridge with this epithelial cell, secrete these granules, and induce apoptosis of this epithelial cell. So turning our attention now to B cells and how B cells get activated and the effector functions of antibodies. As I mentioned previously, free antigens can migrate via the lymph into the draining lymph node, and these free antigens can interact with B cells within the primary B cell follicle. B cells can respond to many types of molecules, lipids and sugars and nucleic acids. Protein antigens, however, tend to be induce the best antibody responses, and that's because only proteins can also mobilize helper T cells. So here's a B cell that's being activated by a virus. This induces some limited clonal expansion of this B cell, and this can lead to the formation of a short-lived plasma cell response in which these plasma cells are producing early IgM antibodies. And so in this way, B cells can make early antibody responses not only to viruses, but also to non-protein antigens. This is all independent of T cell help. However, when activated B cells also interact with T helper cells, these T helper cells produce cytokines, and they express this co-stimulatory molecule CD40 ligand, which in turn binds to CD40 on the surface of B cells. This leads to much stronger B cell activation. So this is a co-stimulatory or signal 2 interaction that enhances B cell activation, which leads to robust clonal expansion, and this is what is required to get the formation of a germinal center. And within the germinal center, some very important things happen. First, obviously, you get a robust B cell response with lots of antibody production. This also leads to the formation of memory B cells. These memory B cells are mostly B cells that produce IgG or IgA or IgE rather than IgM. These memory B cells are capable of rapid recall if they ever see their antigen again. And multiple exposures of these memory B cells to their antigen will boost their ability to make very high affinity antibodies. Within germinal centers, you also can get the formation of long-lived plasma cells. Again, these are mostly producers of IgG, IgA, and IgE. These long-lived plasma cells go to the bone marrow and can persist there for years, and they are continuous antibody secretors. And these antibodies will then exert important effector functions. One of the key effector functions that's mediated by antibodies of all isotypes is neutralization. And during this COVID-19 pandemic, as you've been paying attention to a lot of the talk about vaccines and how vaccines are evaluated, often scientists will refer to neutralizing antibody titers, or how much neutralizing antibody does a person have, and does the antibody neutralize variant strains and so forth. Well, the idea behind antibody-mediated neutralization is that antibodies can bind to the surface of pathogens and prevent their infection, or in the case of certain bacteria in the gut, prevent their colonization or prevent them from secreting toxins that can damage these cells. And so neutralization is a key antibody effector function. A second antibody effector function is opsonization. As you'll recall, I mentioned that complement components are opsonins, that when they coat pathogens, they enhance the ability of phagocytes to recognize and eat those pathogens. Well, the same is true for antibodies. Antibodies can coat pathogens. So here we have a bacteria that's coated with IgG antibodies, can be recognized by a specific class of receptors that recognizes the constant region of this IgG molecule. The constant region is sometimes referred to as the Fc region, and so these are called Fc receptors. This enhances the ability of this macrophage to now take up this bacteria into the phagolysosome where it can destroy it. A third antibody effector function, which is a review of something we've already discussed, is complement activation. Now, I mentioned that complement can be activated by direct recognition of pathogen surfaces, but complement is much more efficiently activated when antibodies are bound to those surfaces. So here is a bacteria bound by antibodies that can recruit complement components that can then lead to the formation of complement structures that induce bacterial lysis. Complement activation can also lead to, again, opsonization. So just like these antibodies can opsonize the bacteria, these complement fragments that are bound to the bacteria will also opsonize it and lead to enhanced phagocytosis. And complement fragments will then also lead to localized inflammation by inducing vasodilation of local capillaries. Now, there are some other key antibody effector functions that we're not going to discuss in this class. We'll hit them up more in host and defense. Don't worry about them for now. That includes antibodies that induce mast cell activation during the allergic response, and antibodies that induce natural killer cell activation during the antiviral response. These are important effector functions, but beyond the scope of what I want to cover in this introductory session. Lastly, I want to end with the concept that antibody responses can be long-lived. A single exposure to antigen with no T cell help can lead to the formation of short-lived plasma cells and a short-lived IgM response. But a single exposure to antigen with T cell help can lead to short and long-lived plasma cells and a long-lived IgG response. And a second exposure to antigens can lead to a massive boosting of this antibody response, the generation of more long-lived plasma cells from memory B cells that are mostly IgG producers with high titers of antibodies with high affinity And these second exposures also boost the number of memory B cells that are available for even further exposures to that antigen. This is the rationale behind vaccination strategies that rely on multiple boosts. That is it for our introduction to immunology. We will discuss these issues at greater length in our team-based learning session.